encounter lots of different writing environments, but it's challenging to measure those because it changes widely depending on location you are in, or time of the year, time throughout the day, and so on. And one simple way to characterizing writing environment may be to measure spectra of individual components in a given scene. So for instance, we can point spectral radiometer to the point of interest like this, and you get the spectra. And in fact, when we think about illumination or writing in general, I think this is what typically implied. We tend to think about the spectra of direct light source, direct illumination, such as sun, or for instance, LED light in my office. But I think our visual experience is much more than that. Light coming from a light source bounce within a scene, so reflects within a scene in very complex way, and those interreflections are also source of our visual input as well. So my idea here is, so instead of characterizing individual elements in a scene separately, why not capture the entire writing environment in a single shot? So this idea of image-based writing, we can preserve all spatial configuration of writing, which might be important when we want to analyze them to understand their influence on our visual functions. So for the first part of the talk, I introduce a technique to measure image-based writing, which stores basically millions of spectra in a given writing environment in a single image. So this first part is about characterizing our external physical environment. Second part, I want to think about our internal world, so our visual system. So so when we are fixating this point, for instance, in a scene, the part of the scene is projected onto our retina like this, and our retinal image, for instance, will be like this. And it's really interesting to me that if you think about our sampling, visual sampling in this way, at a given time point, we are always exposed to this complex spatial pattern of light arrays, which is highly inhomogeneous. So essentially, different parts of the retina are exposed to different spatial patterns of light, which means different parts of the retina might be adapted to different reference points, different white points. And of course, well, this idea is directly inspired by Manuel's conceptualization, spectral diets. So what kind of information do we get in daily life as we live, as we act, not just seeing. So I especially want to think about this spectral diet today from the interaction between spectral diet and our sampling behavior, eye movement. So essentially, I want to think about eye movement as our strategy to standardize light exposure patterns across the retina. So not just thinking about just a one-time point, but we're gonna think about over time, and depending on where you look at, in a scene, different parts of the retina will get different signals over time. So we might be able to find a suitable adaptation point in a given scene by moving our eyes. Okay, let me start with the first point, which is about image-based writing. So measuring natural illumination, it's a big field in human color vision study, and this is because illumination is the origin of inputs to our visual system in daily life. So without the illumination, we don't see anything, basically. So if we can identify how illumination changes throughout the day or depending on location, we might be able to understand the range of visual inputs in our daily life. So daylight measurement has a rich history in color vision research, and one of the most influential studies is probably the one by Jatto et al., which is currently known as CIE daylight model. So what they did, they analyzed more than 600 spectra measured in outside environments at different times in a day in 3 different countries using principal component analysis. And they found something interesting here. So they found only 3 basis functions, just 3 are enough, which means just adding these 3 functions in different ways, we can reconstruct any daylight spectra we typically encounter in your daily life. So natural lighting has a very strong regularity in the spectral shape. And this means if we plot chromaticity of those spectra in a chromaticity diagram like this, they cluster in a very narrow region in the color space, essentially as a single curve, which changes from blue to yellow. And another study also found that basically similar trend in measurement in Granada in southern Spain. So this trend seems to be consistent across different regions in the world. So in our field, in color vision field, there is a widely held assumption, perspective that illumination comes from above like this, and outdoor illumination is limited around this very small region of entire color space. But if you think about a real world object like this, illumination is coming not only from above, for instance, from one side of the object gets skylight like this, from another direction, object receives a different illumination, for instance, sunlight, and from the ground, it receives interreflection. So if you think about a grid around this object like this, lights coming into each cell in this grid from every direction in the environment might have a very different spectral shape. So what I want to understand is basically this, directional structure of the illumination of a thing. And this idea actually has existed for a while as a computer graphics technique called environmental illumination or light probe. I think it's, this kind of data is maybe familiar to some of you, but for me, when I first looked at this, it was really difficult to imagine why this image is illumination. So let me just expand this a little bit. So again, we are imagining an object like this, and we define polar coordinates where the object is origin, center of the coordinate. Then we want to think about incident angle of the light hitting the object from different directions. And we define the direction of the light incident angle using 2 axes, just 2 axes. One is elevation, and the other one is azimuth. So now in this image, each pixel stores information about the illumination coming from a particular direction towards a single point in the scene. So for instance, light, hitting this point is stored at this point, this pixel, and different pixels in this image basically store light coming from different directions in the scene. So this entire image stores information about the lights hitting this specific object from every possible direction. So this is basically the coordinate of this image-based writing. So this is a efficient way of representing all spectra towards a single point in a scene. And using this, what we can do, for instance, is we can simulate how this object, hypothetical object would appear when you place it in under this environment, because from each direction exactly what kind of light is hitting. So this is useful to create realistic stimuli for experiments, for instance. But the current existing database has only basically 3 RGB channels. This is a problem we wanted to address. So instead, if we want to think about the illumination, it's important to have a spectral information. So we basically went outside and collected hyperspectral light probes. And we wanted to understand how spectral varies depending on incident angle in natural environments. So how could we do this? So we use a hyperspectral imaging system placed here, which can sample 33 channels. So unlike RGB camera, normal camera has 3 channels. Hyperspectral camera has 33 channels, which spreads widely over visible wavelength range from 400 to 720. And then this camera is basically focusing on this mirror sphere. This mirror sphere can reflect light from wide range of incident angle like this. So this is just an example mirror sphere image. And the spatial resolution over the mirror sphere region is about 1000 by 1000. So this single image basically stores information about 1 million spectra. But there are two problems here. So we are seeing diffraction of the imaging system itself, which we probably do not want in the final image. And second problem is angular resolution is low towards the edge. So resolution is highest around the center. And towards the edge, basically, larger angular range in the environment is projected onto a single pixel. So you get a very different spatial resolution towards the edge. So to overcome these issues, we take a picture of this mirror sphere from 4 different angles by moving imaging system. Mirror sphere always stays in the same point in the scene. We just rotated imaging system. Then we got 4 mirror sphere images like this. Then we do some sort of like an, coordinate transformation. So you can think about this as like a globe, and we will unwrap this sphere and transform into world map coordinate, which is shown here. Then we just simply stitched all images together, then created hyperspectral light probe like this. Here I'm showing just a rendered object under this specific lighting environment. This is another example that was captured in school canteen. So this is an indoor environment, and it's interestingly different from outdoor environment. So it's, first of all, it has like a ceiling. So it's much more uniform compared to outdoor environment, but it has a large window. So daylight was coming in from this large window. So basically, I'm going to show you the example spectra later, but in this environment, spectra you encounter is basically the mixture of these fluorescent lights placed on the ceiling and the daylight coming from the window. So in indoor scenes, we might have like implicit expectation that it's directionally, spectra is uniform, but even in indoor scenes, there are interesting variations like this. Of course, it depends on the scene. It has some rooms have a large window and others don't. But in the end, we measured 10 scenes in total. So now let's look at how spectral distribution changes depending on incident angle using these 2 scenes as an example. So what I'm showing here is the average spectra across all pixels in this rectangle. So the light coming from this part of the scene, from the sky, has high energy around short wavelength region like this. And if you look at the horizontally, there is very little variation. So from the sky, depending on the angle, there's little variation. But massive variation is seen if you look at as a function of elevation. So lights from above, they are all like kind of like bluish lights, but lights from below in this scene, it's mainly interrefraction from the grass ground. So that's why their spectral shape is largely modified by surface reflectance of these grass. So that's why it's got a very different spectral shape. And if we plot chromatic distribution of all pixels in this image, we can see that they actually largely deviate from the daylight locus. So what I want to say here is if you look at the illumination in this way, they don't really seem to be limited along the daylight locus. And yeah, so in this plot, each spectrum is normalized individually. So intensity information is basically lost. And I have just added the intensity information by background color. And as you see, there's a clear difference between upper hemisphere and the lower hemisphere. And basically lights from above have a few thousand candela per square meter up to 10,000 candela per square meter, while lights from below is few order of hundreds. So there's a 1 log unit difference between lower and upper hemisphere. Okay, so last thing we did in this analysis, we tested whether we can reconstruct these spectra using CI daylight model. And this is a closest fit we could get. Well, as you expect, well, these spectra are matched in terms of correlated color temperature. For the spectra coming from above, you can basically fit really well. But spectra from below, that's interrefraction. And CI daylight model basically doesn't work well. So one thing I want to note here is CI daylight model is widely used, but it does not account for all variations we observe even in outdoor environments. This is just a enlarged figure. And in the scene, the trend is largely different. And as I was explaining earlier, so one big difference is the presence of basically the ceiling. So there is much less clear separation between upper and lower hemisphere compared to outdoor environments. And overall, the spectral shape is somewhat uniform across directions. And these PT spectra basically comes from the spectrum of fluorescent light. And this bit in the environment has like higher intensity, which basically corresponds to large windows here. And around here, spectrum shape is mixture between daylight and fluorescent light. So it's a little bit different compared to others. And other indoor scenes showed more or less similar trend. Okay, so before moving to the second part of the talk, I wanted to just summarize in our first part. So we measured hyperspectral light probe using a mirror sphere and the hyperspectral imaging system. By the way, there are other ways to measure these light probes. You could, for instance, use fisheye lens, which is presented in different papers, but I'm not gonna cover here. We found basically the variation of spectral shape is probably much larger than we previously thought. And there are systematic difference across type of the scene, for instance, between outdoor environment and the indoor environment. And I wanted to also say all these hyperspectral light probes are freely available in a Zenode repository. So if you look at this 2024 paper, this link is presented there. So if you are interested in analyzing spectra of these type of visual environments, feel free to go there and take a look at the data. And one feature of this data set is that we calibrated each image so that each pixel has a absolute radiance unit. I thought which might be useful for community of Circadian research. Okay, so let me move on to second part of the talk, which concerns about our sampling behavior and the spectral diet, the dynamics of our retinal exposure, and how gaze shifts modulate pattern of visual inputs to our visual system. So, so this is a work I did in Portugal. So that's why I'm showing this picture. This is my favorite city in Portugal, and old town in Porto is known for these very beautiful, colorful buildings. And what I want to point out here is Well, this is an extreme example maybe, but in general, the real world presents, natural environment presents complex spatial and chromatic structures, which means at any given time point in our daily life, individual cones on our retina might be exposed to very different spectra. For instance, cones around here sees yellow of the building. Here it's orange, here it's white, and so on. And this implies that adaptation state on our retina might be spatially very inhomogeneous in daily life. But important thing here is our visual experience is not just a passive reflection of external environments like this, because we actively sample information over time. So when we look at this point in a scene at time point T1, this part of the scene is projected onto our retina, and the retinal image would look like this. At a different time point T2, different part of the scene is projected, and T3 like this. And if there are cones at the same retinal position, but basically they are sampling very different colors over time. So essentially, I'm considering this, pattern of gaze shift as a way as our strategy to standardize light exposure patterns across the retina. So if you're focusing on single point in a scene, cones might be adapted differently. And as we move, so eye movement might be one strategy to globally adapt our retina. And I think this is interesting to think about because depending on this light exposure pattern across the retina, we might want to think about optimizing our visual function differently for different retinal coordinates. And one striking example is shown in mice study. And we know that mice are dichromats, but over the past 20 years, it's been repeatedly found that they are dichromats only in the upper visual field, and the lower visual field is actually monochromat. So if you see the distribution of S-opsin expression, this part of the retina doesn't have S-cone. And recent study measured images from mouse point of view, lots of images from mouse point of view, and found that there is not much UV light from the reflection of the ground. So having two types of cones in the upper retina doesn't give you additional information. So this is a reason these researchers speculated why there's this interesting, striking variation. And I think this may be like example where inhomogeneity of light exposure patterns can have critical effects on your visual function. In this case, mouse is organizing retina in a way that they optimize for visual inputs they receive. But for human, what I want to think about today is, so again, our visual experience is not just a reflection of static worlds. So we want to understand how our gaze shift interact with these spectral diets. So the research question I want to address is, can individual photoreceptors sample global scene statistics over time by moving our gaze? So data I'm going to model today, going to show you today, is collected by my co-authors in this project. And they went out like this. And in outdoor environment, they set a table and a parasol. And they set, chin rest under the parasol. Then they measure gaze patterns in 7 different outdoor environments, which are shown here. And task was free viewing. So for 5 minutes, observers were able to look at any point in the scene. And maybe it's hard to see, but they put like a calibration right around here, so that this was used for matching gaze calibration of gaze points. And observers were basically instructed to look at a certain part of the scene for 5 minutes. And 2 or 3 observers participated in each scene. And after eye movements were recorded, basically hyperspectral images were taken from observers, exactly the observer's eye position. So we have, from the point of observer's point, like we have eye gaze data and also hyperspectral images, which is useful, which basically allows us to reconstruct the retinal image during the 5 minutes measurements like this for each observer separately. And hyperspectral cameras acceptance angle was about 6 degrees. So we can look at only 5 degree diameter region, which is basically looking at the fovea. I'm just simulating the fovea as we move gaze shifts like this. And in these retinal images, we accounted for the cone density difference over eccentricity. So the image is blurred towards periphery region in the image. And in this retinal image, we assumed 1 pixel corresponds to 1 set of LMS cone. And here I'm tracking colors sampled at 5 different locations on the retina, which is shown like this. So center of the fovea is here, and so on. So now what I want to know is whether so you see each cone, each photoreceptor is sampling very different colors over time. But now what I want to know is whether these history of colors sampled by each cone over time is similar to global scene statistics. Okay, so first we make global histogram for LMS cone using the entire scene image. So this is basically hypothetical, it's global statistic histogram. So if you sample very uniformly over the scene, this is what you get. Then for each retinal location, for instance, this lower part of the fovea, we combine cone signals over time and draw a histogram like this. So this is a histogram of temporary accumulated signals, especially always, I'm considering 1 point. And we update the histogram over 5 minutes, which is why I wrote frame 1 to frame X here. Then this frame X is updated over time. So in this example, this is just a sampling of colors from frame 1 to frame 4, but we can do this at using a different end of frame. So frame 1 to frame 4 in this case, but frame 1 to frame 1000 for example, and so on. And of course, at the beginning of measurement, cones don't see many different colors. So these local histograms might look very different from this global histogram. But as we sample more information over time, they will look more and more similar, hopefully. We evaluated the similarity by taking correlation across bins for each cone class. So what we want to look at now is how this correlation develops over time. At the beginning of measurement, it's probably very poor, correlation is low, but over time as cone sample different colors, this correlation increase, which is basically shown here. So this is a correlation between local histogram and global histogram of L cone at the center of fovea and average across 7 scenes. And this is data for 1 subject. And as you see here, at the beginning correlation is low, but it rapidly grows during first 10 seconds and gradually increases towards the end of this 5 minutes, end of measurement point. And at this point, correlation reaches nearly 0.9. So this cone can sample nearly identical information to the global scene statistics. I think it's interesting that the correlation rapidly grows like this. So if you move eyes for 10 seconds, each cone is basically adapted. You could adapt it to global scene statistics. And if you look at a different part of the retina, so this is center of fovea. If you look at a different part of the retina, trends are very similar, although trajectory is slightly different. And upper retina, again, trends are different. Trends are similar, although trajectory is different. And here's all other points. So overall, regardless of retinal coordinates you are thinking about, each individual cone can access global scene statistics over time. This is a case for L cone, but we see similar trend across all cone classes. Okay. So our results suggested single cone experience global scene statistics over time, which means our retinal adaptation state probably become homogeneous over time in 10 seconds or so. And finally, I wanted to think about what this means to our color vision system. And specifically using this knowledge, we evaluated the extent to which local cone adaptation can discount the influence of global scene illuminant. So this is a big question in color vision study. So color constancy describes our ability to perceive, basically cancel out the influence of illumination. And people have suggested one way to do is to adapt your cone. But I always wondered, how could we do this? Because cone doesn't know the spatial average color. So we used gaze measurement pattern to simulate our local adaptation, see if how effectively those local cone adaptation can undo the illuminant change like this. So when scene illumination changes, one way to undo the illumination change is through adaptation to mean color. So basically, element signals are divided by mean color. Element signals in each point on this retinal image is divided by mean color across the scene. And this is not perfect, but normally works very well. And in past literature, this manipulation is incorrectly labeled as von Cleese scaling. But von Cleese's original suggestion was actually these gains for each cone classes should be determined locally. And I think this makes more sense as a mechanism at photoreceptor level, because it's not clear how individual cone can adapt to mean color like this. So this is actually the correct formulation of von Cleese's original suggestion. So individual cones adapt to different color over time. Then we have to subtract illuminant influence pixel-wise rather than globally, like shown above. And if we apply this, if our retina is not homogeneously adapted, we might get this kind of weird artifact. So this part of the scene is kind of like a bluish with the artifact and pinkish patterns around here. But again, our retina can be adapted homogeneously over time. We get a very similar results to global adaptation shown above. So that's what I wanted to evaluate. So we are taking this global adaptation on the left as a ground truth, and I wanted to compare pixel-wise delta E in CECM16 space, then took median value as a metric for the perceptual difference between these two images. So basically, this is just to simply put, this is just a way to assess the effectiveness of local cone adaptation. And this is our adaptation model basically we used. So here, according to the eye movement pattern, we are trying to estimate adaptation state at any given time point during the measurement, using a very simple weighted sum of past retinal images. So we are here assuming that at any given time point, our retinal adaptation is basically the weighted sum of past retinal images. And we assign the highest weight to the most recent experience, like this. And towards the past, the weight decays. We can try different time constants, but I'm showing the example of time constant 10 seconds. And then basically using this adaptation state, we applied a pixel-wise local correction to get a corrected image. And for comparison, I'm showing the image corrected by global adaptation here, based on, so this global correction is based on mean color across the entire image. So here we are assuming our visual system can magically access the mean color and use that as a scale to undo illumination change. So now we can look at how delta E between these two images develops over time. So delta E between local correction and global correction. Okay, so this is average across all observers and across 7 scenes. And what I want to, so that I'm showing delta E value over time. And this is the end of measurement, 5 minutes. And I want to say first, so for the illumination change of 4000 Kelvin, it's a massive illuminant change. So if you don't do any illuminant correction, if you don't do anything and just take delta E value between these two, this value is around 30. So this means that the very first, point we consider it is only after 60 milliseconds after the measurement started. Local adaptation can bring delta E value down from 30 to around 10, which is a massive decrease. Then over time, delta E gradually decrease like this. And for the first 10 seconds, for the first 10 seconds rapidly, and again, it reaches plateau around here and gradually moves around like this. So, but the question is, how big is this difference? So just to give us more concrete sense, these are lines of generalities I estimated. So after 10 seconds of adaptation, the difference between local and global correction is less than 2 generalities, which I consider is low. So just after 10 seconds of local adaptation basically gives us perceptually more or less similar experience as global adaptation. Okay, so I wanted to summarize second part. In this part, we looked at the effectiveness of our local cone adaptation to discount influence of illumination and how individual cones can sample global information over time. And we found basically eye movement seems to be an efficient strategy to standardize light exposure patterns on our retina. And I want to point out that, of course, we have cones is just a very first stage of our visual system. And later we have post-receptor system that can spatially compare cone signals over wide spatial region. So it's possible to take spatial mean there, and maybe you can use that as a scaling factor to do global correction. But I want to say, what I want to say is, even at the levels of cones, individual photoreceptors can access global statistics, if only in like 10 seconds or so, which is interesting to me. Okay. Finally, I want to acknowledge my collaborators and funding bodies. So basically, this is a work so for both of the projects, I collaborated with researchers in Portugal. And for the second part of the talk, so Sergio Nascimento and Yao Rinhares and Hannah Smithson. And for the second part of the talk, additionally, I worked with David Foss and Kinji Amano at Manchester. So I wanted to thank their contribution present to the project. And I want to say thank you for your attention.